Okay. Can you hear me? Can you hear me? Yes. Okay, thank you. Uh, okay, finally. All join. Arshad. Yeah, okay. Okay, what we discussed, final part in the, now we discussing the cell structure. In cell structure lesson, we discussed last day what are the cells and cell organelles, cell types. Last day, large part we completed, bulk part completed up to 13 page completed, up to 13 page. Okay, I'll open my PDF. Okay, this is a part. This is a today part, characteristic of microorganisms. Is there any problem? Is it clear? The board is clear or not? Board clear or not? It's clear, no? It's well clear. Okay. Now, today, I'm going to complete the full part of the microorganisms. Actually, the microorganisms are the, this part is the most important and bulk part of this lesson. After that, we have plants, plant tissues completed last day, meristematic tissues and permanent tissues. I think you can remember the classification of plant tissues. Plant tissues mainly two types, meristematic tissues and permanent tissues. Meristems, again, three types. What are the three types of meristem tissues? Do you remember? What are the three types of meristematic tissues? Now, last day lesson, I'm asking. What are the three types of meristematic tissues? Uh, Deneth, what are the three types of meristematic tissues? Three meristematic tissues. Apical meristem, intercalary meristem, and lateral meristem. Very good. Yeah, all are correct. Then what are the different, yeah, permanent tissues? All again two types: simple permanent tissues and complex permanent tissues. Simple permanent tissues mean the cell, same type of cells. The structure is simple. That's why we called as simple permanent tissues. What are the simple permanent types? Simple permanent tissue types. Refresh last day lesson before we go to microorganism. That's why I asked. That's the final part we discussed. Simple permanent tissues, what are the types? Yes, parenchyma, sclerenchyma, and collenchyma. remember them, and they are characteristic and they are cell structures, everything. They are O-level lessons. They are simple. If you refresh last day, that enough. Okay, you don't need deep knowledge, because from these bio lessons, no any essay questions are coming. Only 5 MCQ and 1 structure. Okay, approximate 5 plus 5, all 10 marks from 100. 10 marks easily you can score. Anyone everyone can score this 10 marks. Okay, these easy questions are coming. They are the most easiest question. No any calculation. Only you need to write down answers into the point. Okay, now today we going to start microorganisms. Okay, can anyone send me what is the definition of the microorganism? What is the definition of the microorganism? How you can observe? Last day I explained. Yesterday I explained. What is the definition of the microorganism? The definition of the microorganism? Do you know? Anyway, last day I explained. Few questions we discussed about microorganism. In question discussion, I explained about microorganism. How you can normally the microorganisms they are micro size. The name also given, the microorganisms. It is they are microorganism. That means their size is micro size. Their size is micro. Sometimes their size is nanometer size. Nanometer size. I think you have idea about what is a micrometer and nanometer. We discussed in the measurement lesson. The micrometer means 1 meter divided into 1 million. That's I think you can remember. Micrometer mean 1 meter. 1 meter divided into 1 million. 1 million times. Then one part is a micrometer. 1 micrometer. 1 micrometer. 1 meter, if divided into 1 billion, I think, 9 zeros. That's a 1 nanometer. 1 nanometer. 1 nanometer. 1 nanometer and 1 micrometer. Do you know different types of microorganisms type? You already know. Last day also I explained. Bacteria, different types of microorganisms. Bacteria, normally 5 types. Bacteria, fungi, virus, protozoa, algae. They are the microorganisms. Do you know bacteria and fungi? They are diameter. Their cell sizes. Their organism sizes. It is like 200 to 300 micrometer. 200 to 300 micrometer. Last day also I told about cells lesson. It is 200 to 300 micrometer. Okay, they are comparatively large in size comparative to these viruses. The virus size is like 20 to 30 nanometer. 20 to 30 nanometer. You can see they are different. They are different. If we compare, if it is the virus is like this, if you observe the bacteria, if you magnify the bacteria is like this. Okay, they have compared, the size is totally different. Size is totally different. Okay, however, they all size is less than 0.1 millimeter. Less than 0.1 millimeter. Less than 0.1 millimeter. Okay, last day also I explained the minimum distance or minimum size that we can observe our naked eye, that is the 0.1 millimeter. Okay, 0.1 millimeter is the minimum size that we can observe by our naked eye. naked eye mean normal eye, without any instrument or equipment. Okay. Is it asking, isn't it 300, 400 micrometer bacteria size? Yeah. Yeah, what's the problem? Is it, I can't understand your question. No, it is depend. The bacteria is not only one organism type. Bacteria again different types. Again different types. Normally we can get 200 to 400. Some bacteria maybe 600. Some types maybe 800. Common ranges. Last day I explained. Therefore, the size you don't need to remember. You should have some idea about their common size. Okay, therefore we can't say all microorganisms within this 200 to 300 micrometer. Okay, commonly there are lot of microorganisms, lot of groups within this 200 to 400 range. හරිද? ඊට වඩා වැඩි අයත් ඉන්නවා සෑහෙන. අඩු අයත් ඉන්නවා. The common range is, the average value is 200 to 400. We can get 200 to 400. You don't need to remember that size. Size is not asking in the exam. You need to remember the microorganism, all types of microorganism. The microorganism meaning they can't, organism without observe, the organism can't observe in our naked eye. Can't observe in our naked eye. Their body size is less than 0.1 millimeter. That's a requirement. That's a requirement. Then you can define the microorganism, organisms less than 0.1 millimeter diameter and cannot observe in our naked eye. That's a correct definition for microorganism. Therefore, you can remember 2 points. 2 points. Organisms, they are cell size or organism body size, body diameter less than 0.1 millimeter. That's why they can't observe by our naked eye. Organisms, they can't observe in our naked eye. Their body diameter is less than 0.1 millimeter. That's the definition of microorganisms. Okay, these 2 points you can mention as the definition. Okay, organisms less than 0.1 millimeter diameter and can't see our naked eye. Can't see our naked eye. Okay, very primitive and simple definition that microorganism cannot be seen with the naked eye. When the discussing them, it essential to an adequate understanding of their micrological, physiological, and molecular biological characteristics. There is no ecosystem. This one is very important. If we consider about these microorganism types, as I explained, microorganism type bacteria, fungi, virus, algae, and protozoa, they can live in every environment in the earth. Every environment. You know, every environment mean, if we consider about hot springs, other organisms like animals and plants cannot live. They can't adapt for this hot environment, the high temperature. But microorganism can live even this environment. If we consider about the polar region glaciers, within glaciers, the temperature is minus 18, minus 10 to 18. The temperature is minus 10 to 18. Any plants or animal cannot live, but some type of microorganism, they can tolerate, they can adapt to this environment, and they can live even inside these glaciers. Okay, they are. Do you know in high acidic condition, that we called as abnormal environments. Okay, abnormal environments. The normal plants and animals, normally organism, mainly three types, animals, plant and microorganism. Other any animals or plants cannot live, but only microorganism can live. Therefore, we call there is no any ecosystem in biosphere. What's the meaning of biosphere? Do you have idea what's the meaning of the biosphere? Biosphere can magat. Biosphere we discussing in the final lesson. What's the meaning of the biosphere? Okay, sorry. Anyone, what's the meaning of the biosphere? Biosphere? Sadeep, yes? What's the meaning of the biosphere? Biosphere? This one? This word? Biosphere can magat. You know, Earth, we can divide into yeah, answers. Land, water, air, we. Very good. Sadeep, correct. The zone of life on Earth. Yes. You know, the Earth we can divide into three spheres. We discussed in the final lesson. Three spheres. What are these spheres? Three parts. Think this is the Earth that we called as lithosphere. Lithosphere. Then atmosphere. Atmosphere. And hydrosphere. The water surfaces we called as hydrosphere. 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 Okay, there are three. This is a entire Earth. The lithosphere, atmosphere. Lithosphere means the ground surface. Okay, the water surfaces, hydrosphere. The air surfaces, atmosphere. Okay, there are three surfaces, three spheres. Now every spheres, the living things available. That's been plant, animal, or microorganism living environment, living areas we called as living areas. If we collect, think this area is among these all spheres, the organisms can live, organisms survival. Then this is the biosphere, the collecting of living organism survival, all environment, whole area in the earth, that we called as biosphere. Okay, again, about fully we discussed about biosphere in the environmental chemistry, our final lesson in this tute. In the final tute, we discussed everything about this biosphere, lithosphere, atmosphere, hydrosphere, what are the hydrocycles, and hydrocycles, carbon cycle, nitrogen cycle in the atmosphere. What are the cycles? How these atmosphere cycles interaction with the lithosphere and hydrosphere? Everything we discussed. Okay, this is the spheres, and the every areas, the living things survival, living things living areas that we called as a biosphere. Okay, if we consider about the microorganisms, every biospheres in Earth, every biospheres, the microorganism can live. The additionally, examples: soil, air, water, and animal and plants body with the surface and inside animal and plant body, they can live. They are normal environment. They are normal environments. Microorganisms, plant and animal, all organisms can live without any problem. But additionally, they can live in even in abnormal areas, abnormal environment, like uh, polar regions, like inside glaciers, high acidic condition like pH value 1 to 2, acidic condition, high base condition, pH value 13 to 14, like base condition, uh, other than hot springs, high temperature conditions. Okay, even abnormal condition, other organisms cannot live, plant and animal cannot live, but microorganism, some type of microorganism can live in even in this abnormal environmental climate conditions. Okay, I have given some abnormal environmental conditions. Petroleum deposits, I have given here, you can read. Okay, thermophilous microorganisms, they can live in hot springs. I have given examples. Thermophilus aquaticus, which grow in hot springs. They can live in hot springs. Not for all microorganisms. If we get all microorganisms take as a group, then all microorganisms can't live in high thermal conditions, high temperature conditions. But some type, specific type, they can live in hot springs, like abnormal conditions. Pseudomonas species, they can live in polar regions. Polar region mean cold conditions, cold environment, minus the temperature, minus value conditions, they can live. Okay, thermophilus in hot springs, Pseudomonas species, they can live in polar region in high cold environments. Okay. Okay. Then I have given, then if you consider about microorganism, you can understand everywhere, if it is even in normal environment and additionally abnormal environments, microorganisms are living. Okay, but there is one place in our body, no any microorganism. What do you think? In our body, there is a one place, one material, any microorganisms. What do you think? එක material එකක් තියෙනවා, there is no any microorganism in our body. Amar, what is the where microorganism cannot live in our body? Where microorganism cannot live. If any microorganism enter to this, it is by our immunity system, it is fight about this microorganism, and it is destroyed. Where? Very good. It is in our blood. You know, if we consider our skin more than million type of microorganism, different bacteria type, fungi types, so many bacteria, so many fungi types. That means more than millions of microorganisms can live in our on our skin. But 0.01 millimeter inside blood don't have any microorganism. don't have any microorganism. In our blood and our blood circulation system don't have any microorganism. If any microorganism enter to this blood, what happened? Our immunity system doing fight about this microorganism. අපි අර germ එකක් මේ enter වෙනවා කියන්නේ මේ විස බීජ යනවා කියන්නේ ඒකට. හරිද? If any microorganism enter, it is huge damage. Therefore, the immune system doing their job and destroy this microorganism without their replication. Before reproduction, it is destroyed. Okay, now if we consider only, if we consider about our respiration system, our digestive systems, and everywhere. Yeah, the brain also don't have microorganism. No? Brain also don't have microorganism. But if we consider the brain also the blood circulation, the what we called in brain neurons. Inside neurons don't have microorganism. The neurons also blood circulation don't have microorganism. Okay. But in our respiration system, our digestive system, and other every systems, more than millions of microorganisms. But these microorganisms they can live with symbiotic relationship with us. Not only us, every organisms, their internal system, lot of microorganisms. They doing their living process with symbiotic relationship. Symbiotic relationships mean they are that's mean they are favorable. If you consider our digestive system, for our digestive system, this probiotic microorganism is compulsory. If not, our digestive system is it is not commonly digestive. Okay, therefore for this natural digestive system, the microorganisms are compulsory favorable microorganism. If it is E. coli, salmonella like microorganisms, they are unfavorable for our digestive system. If they enter, what happens? Lot of vomit like problems, fevers coming, disease coming for our digestive system. Okay. Therefore, some microorganisms, some type of microorganism, they doing symbiotic relationship with microorganism and live with other organism inside or on their bodies. Okay, they are the microorganism characteristic. Therefore, if you consider about microorganism, the microorganisms can live in everywhere in the earth. No, they are not environment. Microorganism cannot live environments are very limited. Very limited. Lot of environments they can live. They can live in lot of environments. Okay, what's me now? They are the reasons microorganisms. Now you know they can live in everywhere. They can live in everywhere in the earth due to these reasons. The first reason is they are very small size. That's the first reason. Okay, for one by one we discussed it. They are being very small in size. Therefore, they can live in abnormal conditions as well. The other thing is they have rapid growth rate. This is an exam question. Okay? This is the first point came in the exam. You want to remember the definition of the microorganisms. What is the definition of the microorganism? Organisms can't see in our naked eye, and they are the body diameter less than 0.1 millimeter, and the microorganism can live in every environment in the earth, even in abnormal environments. Now, due to these reasons that you need to remember. Okay, the first reason is they are very small in size. The other one is they have rapid growth rate and rapid reproduction rate. they are growing rapidly and reproductive rapidly. They are produced next generation within very short time. Viruses, they are very rapid growing rate and reproductive rate, having more wide morphological and physiological diversity that we can discuss in cell their organelles discussion. Microorganism, if we consider bacteria prokaryotic structure, prokaryotic structure, their structures, organelles, very simple. The tube. Give me a second. This one. Oh, sorry. Okay, I think you all have tubes now. If not, please send me a message. I will give you the PDF. Otherwise, very difficult to do this lesson because lot of bulk theories here. You all have, no? Only one student I given for him. Okay. Other side. Okay. That's my pen out. Okay. They are the third factor. They have wide variation of the morphological and physiological characteristic. Morphological and physiological characteristic. The other very important thing is they have wide nutritional diversity. Okay, one by one we can discuss after. This is very important. They have main 4 nutritional groups. Okay, that we discussed in the next page. Okay, the other one is in morphological characteristics. They have 4 respiration diversity. The respiration diversity also you can take it as a separate one. They have nutritional diversity and they have respirative diversity. Okay, at this another one. Okay, now they are the factors. The other one, as I explained, they can live with other organism with symbiotic, asymbiotic relationship between other organisms. Other organisms mean animals and plants. With animals and plant, they can live with symbiotic and asymbiotic relationships. Okay, they are the factors. What are the factors? You have to remember this. All factors. They have rapid growth rate. What is the second one? Rapid growth rate, and they are very small in size. They have rapid reproduction rate. And what is the other one? Morphological and physiological difference, nutritional difference, nutritional diversity, respirative diversity, and symbiotic and asymbiotic relationship with other organisms. Okay, all these characteristics you have to remember. Understood up to now? Any doubt up to now? We discussed about who are the microorganisms, what are the environments microorganisms can live, and finally we discussed about what are the reasons microorganisms can live in every environments. They are the reasons. Understood up to now? Understood? Any doubt? Okay, no doubts. Okay, next one, the morphological diversity of organisms. Okay, now we're going to discuss one by one what are these characteristics. Okay, morphological diversity mean, as I explained earlier, some, if we consider about bacteria, they are very simple structure. The unicellular organism, the cell is very simple, don't have organized nucleus, and they have only one chromosome number, as I discussed in prokaryotic cell structure lesson. Last day we discussed every parts and every functions of bacteria, no? Then it is very simple structure. But if we consider about fungi, they are eukaryotic cell structure. Fungi is a eukaryotic cell structure. They have organized nucleus. Now you can see there is diversity between each and every microorganism type. If we consider about viruses, they have totally different morphological characteristics. If we consider about algae and protozoa, they are different from others. Therefore, the diversity important for the everywhere growth in microorganisms. Everywhere live in microorganisms. Okay. Other very important thing is how we observe these morphological characteristic of this morphological characteristic mean they are organelles and internal structure. How we observe? Are they observable in our naked eye? No. It is less than 0.1 millimeter. No? It is less than 0.1 millimeter. Therefore, we can't observe. Therefore, we have to use different types of equipments. Microorganisms are always organisms less than 0.1 millimeter size, remember. This you need to remember. Okay, mainly we can use this light microscope and electronic microscope. If we want to observe only organisms, their internal organelles, we can use, for bacteria and fungi, we can use light microscope. For viruses, we have to use electronic microscope. If you, in bacteria and fungi, if you want to observe internal structure of their cell organelles, not only organelles, internal structure of cell organelles, then we have to use again electron microscope. Okay, who found this microscope firstly? Who found this microscope? Do you remember in O-level science who found microscope? Who is the father of this microorganism? Father of microorganism. Who is this? What's the name of this scientist? And not only enter. Yeah, the Anton van Leeuwenhoek. Anton van Leeuwenhoek. He found the first microscope. After that, invented different types of technologically improved microscope. Then we can observe their morphological characteristic. They have considerable morphological diversity. That's advantage for everywhere living microorganisms. This diversity. Okay, now we come to our next point, the respiratory diversity. Respiratory diversity. According to respiratory diversity, microorganisms we can divide into 4 groups. 4 groups. Okay, you can read these things, not important. Microorganism, this respiration important for their metabolic activities and their energy generations, like us. The respiration, whatever respiration method they must have for their energy metabolic activities. Okay, now microorganism classified into main 4 respiratory groups. They are aerobic, anaerobic, and facultative anaerobic, and microaerobic. Okay, I will explain one by one. They are the exam asking questions. Always ask MCQ coming parts. Therefore, remember very well what is the meaning of aerobic. Okay, aerobic. I will explain in the board. No need, no, we can explain. I will explain here. Okay, aerobic microorganism. Aerobic microorganism mean they, like us, okay, they need oxygen. If it is not oxygen, they cannot live. If don't have oxygen, they only can live in oxygen with environment. Oxygen with environment. If it is oxygen, that's mean anaerobic conditions, oxygen less or oxygen without environments, they are toxic for them. Okay, like us, the aerobic microorganisms, they need oxygen for their metabolic activities. Aerobic mean they absorb oxygen and produce energy for their metabolic activities. Okay, therefore aerobic microorganisms are, they need oxygen for their metabolic activities. Anaerobic microorganism, the opposite side. Anaerobic microorganism mean they only live in oxygen without environment. Oxygen without environment. The oxygen with environments toxic for this microorganism. As example, methanobacteria and pseudomonas, they are anaerobic microorganism. I will give you examples. They are anaerobic microorganism. They only live in oxygen less environment, not oxygen less, oxygen without oxygen environment. this environment, the oxygen should be zero. If not oxygen, they can live. They are anaerobic microorganism. Do you know methanococcus, methanobacter, methano species microorganism we use for biodiesel production. In next part we can discuss this biodiesel production producing bacteria are methano species. They only doing their job in anaerobic conditions. Anaerobic conditions, they can't live in oxygen with environment like that. Me normal environment they can't live. Therefore, they need fully closed environment without oxygen in this environment. Okay, they are for biodiesel production. We need fully closed environment. Fully closed environment. The oxygen amount should be zero. Oxygen amount zero. Okay, facultative anaerobic. Facultative anaerobic, very special type of microorganism. Facultative anaerobic microorganism mean they can live in both environment. Okay, if it is oxygen available or not available, they can live. It is not problem. It's not problem. Okay, that's a facultative anaerobic. If you consider about, do you heard about yeast, Saccharomyces cerevisiae? The Saccharomyces cerevisiae is a facultative anaerobic microorganism. Facultative anaerobic microorganism. Therefore, if it is can anyone send me? This is facultative anaerobic. The facultative anaerobic mean they can live in both environments. Oxygen with or without, both environment they can live. But why we call this as facultative anaerobic? What is the reason? If they can live in aerobic and anaerobic both environments, why we called as facultative anaerobic without saying it is facultative aerobic? Any idea? What do you think? Then we can call this as facultative aerobic කියන්නත් පුළුවන්. What's the reason? Any idea? Yeah, facultative mean optional. It's okay. I'm asking why we not name as why they not called as facultative aerobic. The reason is, if we consider about the yeast or yeast means Saccharomyces cerevisiae. Saccharomyces cerevisiae is an example of this facultative anaerobic. The facultative anaerobic microorganisms, they can live in oxygen with or without both environment, but they more prefer to work on anaerobic conditions. Anaerobic conditions more preferable. Okay, they doing their job very well, very rapidly in anaerobic conditions. If it is oxygen available, it's not toxic for them. It is not a toxic like anaerobic microorganism, but they more prefer to work in anaerobic conditions. Okay, yeast is the example. Then oxygen with or without conditions. Oxygen with or without both conditions, they can live. Okay, microphilic microorganisms. Microphilic. What's the meaning? Microphilic. Microphilic. What do you think? Now you know aerobic, oxygen only with oxygen environment like us. Anaerobic mean oxygen with environment, oxygen available environment. Toxic, therefore it should be zero oxygen environment. If it is oxygen available or not, facultative anaerobic, okay. Okay, what do you think? Who are the microphilic microorganisms? Microphilic කියන්නේ කවුද? Very good. They need less oxygen environment. Less oxygen environment. That means the amount of oxygen 5 to 10%. Remember this percentage. 5 to 10%. 5 to 10% is required for their growth. If it is more than 10%, it is toxic. If it is less than 5%, it's also toxic. Okay, remember 5 to 10% less amount of oxygen environment, less amount of oxygen, they can live. They can live. Only less amount of oxygen with environment, microphilic microorganisms can live. Okay, I will show you a small example. Small example for understand this aerobic, anaerobic, facultative, anaerobic, and finally microphilic microorganism. Okay, I'm going to take 4 water beakers. Okay. They are 4 water beakers. 4 water beakers. Same size, 4 water beakers. Okay, now think we add same amount of water. Same amount of water. Okay, for first one, A, B, C, D. If I add aerobic microorganisms, where this, think it is from A, B, C, where aerobic microorganism, where they prefer to live, where most prefer to live, the upper surface or mid level or down bottom level. Aerobic microorganisms, aerobic microorganisms, aerobic. Yes, they always prefer to live in this upper surface of the water because it has more oxygen. Oxygen is higher than other bottom areas. Therefore, always they prefer to go for upper surfaces. Upper surfaces. What's the reason? They can easily take oxygen. Easily take oxygen. Okay, if we add anaerobic microorganism into B test tube, where they live? Where they live? Not live. Actually, they anywhere they can't live because it has water contains some oxygen level. Where they prefer to live? Bottom or up or mid level? Yes, they go to bottom areas because in this area the oxygen value is very low. Oxygen value is very low, oxygen percentage very low. Therefore, they always try to go for maximum bottom surfaces. Okay, if we consider about facultative anaerobic. Facultative anaerobic microorganism, if adding, they can live in not only mid level, they can live in any environment, but they prefer to live in the more concentration, they prefer to live in bottom. They can live in any environment. They can live in anywhere in the water surface. They can live in anywhere, but more prefer to go for bottom areas. More prefer, but in upper surface also they can live. They can live not toxic condition. Okay, if I add microphilic microorganism into this 4 test tube, where they can live. Yes, good. In bottom is anaerobic. Think bottom is anaerobic, no oxygen in bottom. Then think when increase the depth of the water, the oxygen level is gradually decreasing and the bottom no any oxygen available. If we consume like that, where they can live? Dinit, Arshad, what do you think? In upper region or bottom region or mid? Amavi, what do you think? A lot of answers, different answers. Consume. The bottom is no oxygen. The upper surface, lot of oxygens. Then when increase the depth of the water, when increase the height, uh, the oxygen level is decreasing. The microphilic microorganism, yes, they always try to live in this area. They need oxygen, but normal environment they can't live. You know, in normal. Okay, now okay. Give me a second. Okay, sorry. Now okay, no? Now, okay. I told in environment, normally more than 20%, approximately 21% of oxygen concentration. Therefore, this concentration is toxic for these microphilic microorganisms. Microphilic microorganisms. Therefore, they only need 5 to 10 specific less amount of oxygen. But if it is anaerobic condition, again it is a toxic condition for microphilic. Okay, therefore 5 to 10% they only can live in this area. This much of height from the upper level. Not mid mean normally 5%, more than 5% they need. More than 5% they need. Okay, then you can remember, I think. Okay, aerobic microorganisms. Write down in the aerobic microorganisms. Microorganisms can live in only oxygen available environments. Microorganisms can live only in oxygen available environment. If oxygen not available, if oxygen not available, it is toxic for aerobic microorganisms. If oxygen not available, it is toxic for aerobic microorganism. Write down examples. Acetobacter. Acetobacter species. Acetobacter species mean Acetobacter group. It's a group, not only individual microorganism. It's a group. Acetobacter species. Acetobacter species. Acetobacter species and gluconobacter. Gluconobacter species. That enough. Gluconobacter species. Acetobacter, gluconobacter. They are aerobic microorganisms. Without oxygen, they can't live. Without oxygen, they can't live. Only if oxygen available, they can live. If not available, they can't live. Understood? Okay. Anaerobic microorganism, write down. Microorganisms can only live in microorganisms can only live in without oxygen conditions. Without oxygen condition. Oxygen available environments are toxic. Oxygen available, O2 available environments are toxic. Write down examples. Methano species. Methano species mean methanobacter, methanococcus, methanobacter, methanococcus, there are 4 types methanobacteria. That's all groups we called as methano species. And Clostridium species. Clostridium species. Clostridium species. Okay, that enough. If you remember 2 examples, that enough. Okay. Facultative anaerobic microorganism, write down. Oxygen with O without both environments. Favorable for living. Oxygen with O without both environments are favorable for living. But more prefer to live in anaerobic conditions. Examples Saccharomyces cerevisiae. Yeast is that enough. Saccharomyces cerevisiae is facultative anaerobic microorganism. Microphilic, write down. Microphilic. Microorganisms only live in less concentration of oxygen level. Only live in less concentration of oxygen level. Within bracket 5 to 10% oxygen level. Within bracket 5 to 10% oxygen level. 5 to 10% of oxygen level. Second point, higher concentration or lower concentration. Higher concentration or lower concentration than this range, higher concentration or lower concentration than this range are toxic. Are toxic. Are toxic. Examples: lactobacillus. 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 It is SCC. Lactobacillus. 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 And there is another one, Campylobacter. Lactobacillus species. Lactobacillus species. Campylobacter. Campylobacter species. Remember, Lactobacillus and Campylobacter. Lactobacillus and Campylobacter species. Okay, they are the microphilic or microorganism. Okay, now do you remember where we use these lactobacillus microorganisms? In which industry? Lactobacillus microorganism where we use? In which industry? Where they use Karan? Lactobacillus microorganisms? Where we use? In dairy productions. Good. Yogurt, curd production we use good Amar. In dairy productions we use this lactobacillus microorganism. Now you can get idea if you use, I think now that's why I given most common and most popular examples. Okay, now you already know these old examples. Okay, now if you need, okay, I will show you this acetobacter we use in vinegar production. Next in the production process, acetobacter we use for vinegar production and gluconobacter both. Okay, therefore we need to give more favorable, more suitable, favorable condition. We need to give more oxygen for them. Okay, more oxygen contaction is requirement for aerobic acetobacter in vinegar production. You know, anaerobic, the methanol species and clostridium we use for mainly methanol species we use for biogas production, for energy produced from organic materials. Then we need to maintain which condition? Full anaerobic closed conditions. Okay, now you have idea. Okay, facultative anaerobic, the yeast always we use Saccharomyces cerevisiae. Then you have to maintain which condition? If we can maintain anaerobic condition, they are more preferable. Therefore, for they doing fermentation process, if we can maintain anaerobic condition, if they have oxygen, they are not toxic. For them, it is not a toxic condition, but they more prefer to live in anaerobic condition. Anaerobic to na that. Then microphilic lactobacillus campylobacter we can use for dairy industries, dairy productions. Therefore, is it okay for open environment? No, we need to close and give limited amount of oxygen, less amount of oxygen for their incubation. Incubation means favorable conditions. Understood? Any doubt in respiratory diversity? Quickly. Understood? Tell me all understood or not. What's your comment? Okay, good. Okay, now that is the respiration diversity. You can make a small short note. Okay, respiration diversity, the respiration method, actually the very simple, don't need short note. If you for exam, if you want, you can small short note what are the respiration group and which environment they prefer to live and what are the examples. Okay, nutritional diversity, the other very important thing. Okay, we discussing the microorganism, what are the reasons microorganisms everywhere in the earth, they are the reasons, one by one we discussing. Okay, get the idea. First of all, we discussed about their small in size, the sizes we compared. Then we discussed morphological diversity, respiration diversity. This is the nutritional diversity. Nutritional diversity. Okay, mainly according to nutritional diversity, also microorganisms we can divide into four main groups. Four main groups. They are, you need to remember this classification mainly depend on the microorganism carbon source and the energy source. Carbon source and energy source. Their carbon and energy source mainly important for this classification. Okay, according to their carbon and energy source, we can divide microorganism, all microorganism into, we can classify it into four main categories, nutritional groups. They are photoautotrophs. In, I think in last week exam question I explained photoautotrophs, photoheterotrophs, chemoautotrophs and chemoheterotrophs. Very simply to remember, photo, two photo and two chemo. Autotropes and heterotrophs. Autotropes and heterotrophs. Okay. Okay. If I fill this, okay, first of all, fill this table. That's a short note. Then you can complete this note. Actually, I'm not going to give this note. I'm only give the carbon sources and energy sources. That enough for your exam. No, if you have this. If you remember this table, that enough for exam. Any MCQ question you can do. Okay, now okay, nutritional method. First of all, first one is autotrophs. 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 Okay, photoautotrophs. You can remember this everything with these terms. These terms. Okay, do you know? You already know what is the meaning of photo. What's the meaning of the photogenic? What's the meaning? Photosynthetic. Photo mean, yeah, the photo is the light. Mainly where it is light coming from sun. Then the photosynthesis are, remember, their energy source is sunlight. Their energy source is sunlight. Autotrophs mean, what's the meaning of autotrophs? Auto, okay. What's the meaning of autotrophs? Autotrophs කියන්නේ කවුද? Yes. Others? Who are the autotrophs? Autotrophs. Quickly. Who are the autotrophs? Isuru. Very good. Those who produce their own food. Very good. Then if they autotrophs, they produce foods. Then for produce food, they need to photosynthesis process. Then for photosynthesis, what they need? Carbon dioxide required. Then carbon source is carbon dioxide, remember. Then the examples are green sulfur bacteria. That means cyanobacteria types, okay. Green sulfur bacteria and purple sulfur bacteria. 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 They are examples for autotrophs. Very simple. Now the second one, heterotrophs. Photoheterotrophs. Then they have photo. Then photo mean they use sunlight. Don't need to think. Sunlight they using. But they heterotrophs. Heterotrophs mean they cannot produce their own foods like us, animals. They cannot produce their own food. Then what is the meaning of what is their carbon source? Remember, their carbon source is they need to get foods like us. Their carbon source is organic matters that we called as organic carbon. No, carbonic carbon. So organic carbon. How we call this? Yes, organic carbon. Organic carbon. Organic carbon. That's mean organic matter. They have to depend on organic matter because they are heterotrophic. Don't have chlorophyll. Remember examples. The opposite side of phototrophs. Non-green sulfur bacteria. Remember examples. Non-green sulfur bacteria. Non-purple sulfur bacteria. Non-purple sulfur bacteria. Non-purple sulfur bacteria, non-green sulfur bacteria, they are the example for photoheterotrophs. Okay, third one, chemoautotrophs. Chemoautotrophs. Okay, what do you think? They are autotrophs. They are autotrophs. That's mean they produce their own energy. Therefore, but they not use photo here. Photo means they not use sunlight. Therefore, it is keep empty, and the carbon source is carbon dioxide for photosynthetic process. For their food production, carbon dioxide required. But they not use sunlight. They use different chemicals that we called as chemoautotrophs. They produce their own food by using different chemicals. Therefore, the chemicals that we called as inorganic compounds. Inorganic compounds. not organic, inorganic compounds. Inorganic compounds. Who are the example? Last week exam question came from this point. Who are the examples? Do you remember? Chemoautotropes examples? They are common group of bacteria. Common group of bacteria. What's the name? Do you remember nitrification bacteria? Nitrosomonas, nitrobacter? Nitrosomonas. Remember these lines. Nitrosomonas, Nitrosomonas line and nitrobacter. Nitrosomonas and nitrobacter. They are the example. They we called as nitrification bacteria. About nitrification, I will explain in the next page. Okay, final one. The final one is chemoheterotrophs. 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 Who are the what is the energy source? The energy source now they use chemicals. Therefore, inorganic compound. Inorganic compound. Inorganic compound. Heterotrophs. Therefore, they cannot produce their own foods. The heterotrophs, they are organic compound we use for the organic compound used for the use as the organic carbon. Organic carbon used as the carbon source. Who are the examples for this one? Final one, chemoheterotrophs. Yes, animal like us, chemoheterotrophs. No, I'm asking about microorganisms. We're discussing about microorganisms. Yes, all fungi, very good. All fungi. What else? Protozoa. What else? The main one you missed. What's the main one? That's why I keep the space. Who are the main microorganism? Chemoheterotrophs. Bacteria, very good. Without cyanobacteria, all bacteria that we called as heterotrophic bacteria. Heterotrophics. Heterotrophic bacteria. Heterotrophic bacteria. Okay, that's a full nutrition diversification. Nutrition diverse classification according to carbon and energy source. Okay, quickly complete this table. This note you can complete according to okay, if you want, I'm not going to give you the same thing I want to give you. The autotrophs mean organisms, microorganisms. The carbon source is carbon dioxide and energy source is using as the sunlight. The examples you can write down. Okay, now you can prepare notes in your own. Okay, complete this table. That's a full summary of nutrition diversification. Nutrition diversification. Okay, any doubt from nutritional? Are you complete? Quickly, are you complete the table? Then that enough. You can make later the notes. Okay, actually not compulsory. That's only you need to know. Complete? Or do you need more time? Okay, question paper, sorry, page number 16 complete. Now we move into now 8:30. Now we move into our microorganism related productions. Okay, if we can complete all production today, including bioremediation and wastewater treatment, and finally biogas production, then 50% of this lesson is complete. Okay, complete, guys. You complete, send me yes, otherwise I don't know. Okay, good, very good. Okay, now we move into microorganism related productions. Microorganism related production, how we utilize this microorganism for technological productions actually. How we use this microorganism for technological productions. How we use microorganism for technological productions. Okay. First of all, we're going to discuss how these microorganisms important for agriculture. You know, as we discussed earlier, the microorganisms doing a very important job in soil fertility management. The soil fertility management and the soil structure development, microorganism doing a huge work. You know, the soil is if you consider about the structure of the soil structure is like this. They are like, a soil is like crystals, no? Like this. between the soil individual crystals, they have some gap between each other. What they do, these microorganisms important to produce the structure of the soil. Structure mean the organic material on the soil. By microorganism, they degrading. After degrade, what is produced? The special compound, special black color crystals are coming. The name is humus. This humus very important for this soil fertility management. Normally, that we called more than the mass of the humus can retain more than 3 times mass of water from their weight. As example, think one humus is 1 gram. If it is 1 gram, that can retain 3 grams of water. 3 grams of water that can retain in the soil. I told between the soil particles, soil crystals, lot of spaces that between this each and every soil particles has spaces. These spaces, what they doing? These spaces, between these spaces, this microorganism produce humus between these spaces. Think this bold color one is humus. Then what they can do? They can retain more water in the root system, in the upper surface of the soil. If not, think this is the upper surface of the soil. After rainfall, what happened? The water is percolating to the ground layer. The percolation means the water go to deep layers of the soil. That's the meaning of percolation. Okay, percolation. The water go into deep layers through the soil. That's the percolation. Okay, if it is not any humus in the soil or organic matter in the soil, all water coming from rainfall go to deep layers, and in root system no any water, and therefore when it is available humus, what they can do, they can retain some water amount in the upper soil surfaces. Therefore, and they can make the structure of the soil and manage the soil fertility. Soil fertility means when organic material in the soil, when it is decomposed, it's producing humus. That humus contain balanced nutrition value. Balanced nutrition value means for plant development, the main nutrition requirement is NPK, that we call NPK, nitrogen, phosphorus, potassium are the main nutrition required for plant growth. They are that we called as micro nutritions. No, macronutrients. The micronutrients required in small amount. Macronutrients. The nitrogen is the most required nutrition. Phosphorus next. The potassium next. Therefore, this organic material decomposition, the humus production process that release balanced nutrition, NPK, balance according to plant requirement, they can enter to the soil and mix with soil by this organic material. If we consider about the forest area for plant development, do we need to add fertilizer additionally? Do we need add chemical fertilizers or whatever organic or whatever fertilizer we adding? Are we adding? Yes, we're not going to adding any fertilizer, whatever chemical or organic, whatever fertilizer we're not adding. what they're doing in forest or whatever the natural environments, but they, how it is then soil fertility is decreasing. No, with organic material, the plants parts and dead plant parts in the plants, they decompose by microorganism in the soil, then the humus producing, then this humus provide balanced nutrition for the plant development. Therefore, no any deficiencies, nutrition deficiencies, and the balance, the soil fertility is very high in this area. And if you go and if you check the water level and the moisture level of this fertility soil, you can see, I think if you have observed, the in forest area, the soil, the upper layer soil has more moisture level. The reason is this humus contain more moisture. They can retain more moisture in the upper area of the soil. Are you got the point? Therefore, the microorganism very important to soil structure development, water retention, and soil fertility management of agriculture field. Are you got? These things you need to understand. You can't by heart and keep these things. If you understood, you can remember this. Okay, send me this story is okay. That's the main thing doing in the soil by microorganism for agriculture. For agriculture. Okay, the other very important thing is by microorganism, this nitrogen, you know, this for agriculture, the nitrogen is the main fertilizer deficiency coming in nitrogen. The reason is a lot of plant parts, when we removing as the yield, the lot of nitrogen is removing from the vegetative field. Therefore, the nitrogen is the most critical nutrition in the vegetative field. Therefore, but if we consider about atmosphere, the nitrogen concentration, it is approximate 76%. No? Therefore, no any problem. Therefore, the 76% is nitrogen. What is the problem? This nitrogen gas, N2 gas, cannot use for plants. If it is oxygen, they can use, they can absorb by the respiration for plants. The problem is nitrogen, no any method to absorb nitrogen as the N2 gas. N2 with the 76% but more than 76, no? 76 point value. Then plant cannot absorb any nitrogen gas. Therefore, for nitrogen requirement, the nitrogen for nitrogen requirement, nitrogen need to convert into NO3. NO3 mean nitrate. NO3 mean nitrate. If it is convert into nitrate, only plants can absorb. The nitrate is an ion type. The nitrogen gas is not dissolved in the water, but this the ion is dissolved in the water. Then with plant absorb water, these nitrate ions can absorb to the plant. That we called as the nitrogen, not plant available nitrogen type. The nitrate is the plant available nitrogen types. The plant available nitrogen type is nitrate ions. Okay, for this conversion process, nitrogen convert into nitrate. This conversion process that we called as nitrification. This conversion process we called as nitrification. This conversion process we called as nitrification. Nitrogen nitrate conversion process we called as nitrification. Okay, after it's produced nitrate, this is aerobic condition. Nitrification is aerobic condition. Remember, nitrification doing by aerobic bacteria. For this process, microorganism very important. This is doing by microorganism process. Okay. And if it is anaerobic condition, this is aerobic condition, remember. Aerobic condition. Aerobic condition. That means they need oxygen for. If it is no oxygen, this bacteria cannot live, that cannot survive. Therefore, the nitrification is not happen. It's not continuing. Okay, if it is anaerobic condition, remember this nitrate ion again convert into nitrogen gas. Nitrogen gas. We can consider this as a cycle. See, the nitrogen gas convert into nitrate ions in the soil. This is soil. Then again, in an anaerobic condition, again it convert into N2 gas. Now you can see it is like a cycle that we called as a nitrogen cycle. Nitrogen cycle. Remember, this is nitrification. This is nitrification. This is nitrification that we called as denitrification. This is nitrification. That is the denitrification. The nitrification and denitrification both we called as nitrogen cycle. This is a 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 nitrogen cycle. The nitrification and denitrification. The nitrification and denitrification, that nitrogen cycle, very important for keep the nutrition balance of the environment and soil. For this both process, nitrification and denitrification, both microorganism very important. Microorganism very important. If we consider about nitrification, for this nitrification, aerobic microorganism compulsory required. Aerobic microorganism. For denitrification, it is always happening. The nitrate nitrogen conversion is happening in anaerobic conditions. Anaerobic condition without oxygen. Therefore, that is mainly doing by pseudomonas like anaerobic microorganisms. Anaerobic microorganisms. Okay. Okay, I have given in the page number 18. The nitrogen cycle I have given. Before that, I have given few information. You can read this. Okay, read the nitrogen cycle. Read the nitrogen cycle. Give me a second. Read the nitrogen cycle. Yeah, good question. Good question asking the ammonification. Yeah, Arshad, the ammonification is a part of the nitrification. The ammonification mean ammonium producing. Okay, I will explain now. Okay. Okay, good question. Okay, now I'm going to explain simply the nitrogen cycle, how involve microorganism for the nitrogen cycle. It is not fully nitrogen cycle. The full nitrogen cycle we discussed in the next lesson. The nitrification is denitrification. That's only that the denitrification only doing by microorganism, anaerobic microorganism. But the nitrification is happening due to three methods. If you go to environmental chemistry, get this tube and go to environment tube, page number 5, this one. Then I have given here the nitrification. Fully I have given here the nitrification. You can read, and I will explain here how microorganism involved for this nitrogen cycle, that we discussed in the cell structure lesson. The nitrogen cycle fully with other method we discussed in the environmental chemistry lesson. If you want, you can read here. The nitrification we can divide into mainly three types. Okay, nitrification mainly three types. Okay, come to nitrification. This nitrogen cycle is, I think, this time most chance to come nitrogen cycle as a structured question. Okay, therefore remember, because a lot of microorganism involved for this process. Last time also no nitrification question. Okay, the nitrification. Nitrification nitrification is nitrogen converted into nitrate. Okay, nitrogen converted into nitrate, convert into nitrate. This is the plant available nitrogen type nitrate ions. Now you already know the nitrogen fixation or nitrification. It is three main methods. The first one is biological fixation. Biological fixation and atmospheric fixation and industrial fixation. The nitrogen cycle, the nitrification process, we can divide. There are three nitrification methods mainly: biological fixation, atmospheric fixation, and industrial fixation. But microorganism involved for this biological fixation. Some microorganisms, they can freely live in environment, freely live in the soil, like Acidobacter I have given here. Acidobacter, Anabaena, and Clostridium bacteria, they freely living in the soil, and nitrogen gas they convert into nitrate. Nitrogen gas convert into, not actually nitrate directly, they directly producing ammonia. The biological fixation mean whatever freely available or symbiotic bacteria. symbiotic mean rhizobium like microorganism. They live with symbiotic relationship with plants like leguminous plants. Leguminous mean leguminous, I think you know. What's the meaning of the leguminous plants? Leguminous plants කියන්නේ මොනවද? Leguminous plants. Yeah. What are the examples for leguminous plants? Leguminous mean it's a plant group. Leguminous. 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 What are the example? Only. What are the other examples for leguminous plants? Amar, what are the examples for leguminous plants? Normally, this rhizobium bacteria is living with symbiotic relationship in leguminous plants. Leguminous mean just beans like in Sinhala Ranila Kuleshakya, like they produce root nodules. Root nodules hadala, they can live in symbiotic relationships. Okay, the leguminous plants. Okay, now this whatever Acetobacter or leguminous like leguminous plant available rhizobium bacteria or nitrogen gas in the atmosphere, they convert into NH3 ammonia. Okay, ammonia, they convert into ammonia. They cannot directly nitrification, but they can help. That we called as a nitrogen fixation. Nitrogen fixation is not the full nitrification. The nitrogen fixation is a part of nitrification. That we called as ammonification bacteria. I got that? That is the ammonification. Ammonification mean whatever microorganism living in the soil freely or symbiotic with other organisms, they convert nitrogen gas into ammonia. Okay, they can convert nitrogen into ammonia. That's the meaning of nitrogen fixation. Nitrogen fixation. Okay, that is a biological nitrogen fixation. That's a biological nitrogen fixation, freely available microorganism or symbiotic microorganism. Okay, atmospheric nitrogen fixation. What's the meaning? It's no more atmospheric nitrogen fixation. You can read like thundering. Thundering is a process. In thundering, this due to this energy, atmospheric nitrogen directly convert into ammonia. Okay, that's also a nitrogen fixation process, but no more microorganism required. The industrial fixation mean with some ammonium fertilizer adding for in different industries. Therefore, only biological fixation we required microorganisms. Okay, biological fixation, they are not related to microorganism but parts of nitrogen cycle. Okay, the full nitrogen cycle that we can discuss in the next lesson. Now we going to discuss fully microorganism involvement of the plant nitrification. Make a balance. How nitrogen microorganism involvement for the nitrogen cycle, nitrification. Okay, first of all, you know the plants and animal death material. Plants and animal death material. Death plant animal parts. You know, dead plant animal parts contain proteins, no, that we called as animal and plant proteins. Understand this one, very important. Animal and plant proteins. These proteins, these animal and plant proteins come into soil by dead plant and animal materials. They contain some proteins. You already know the protein contains some nitrogen, no? The protein contain, because every protein is producing with amino acid, then amino acid contain amine group, therefore it is NH2, therefore it is contain nitrogen. Therefore this freely available acetobacter, I previously explained, freely available microorganism like acetobacter, anabina, and clostridium. Acetobacter, anabina, clostridium, like bacteria, what they doing, they break down this organic material, plant and animal dead parts. After that, convert into, break down and convert into amino acid. Simple condition. Amino acid, that's mean convert into amino acid, that means like hydrolysis process. This hydrolysis process, now it is simplest, now it is simple compound. Again this bacteria act on this amino acid, then producing NH3, that we called as ammonification. This is the process of ammonification. Write down ammonification. This ammonium production process, ammonification. Arshad, understood? The nitrogen cycle is a long story. I am explaining this. This is the ammonification. Arshad, I got ammonification. Ammonification can make. Yes, this is the ammonification. Remember this ammonification bacteria, Acetobacter, Anabina, and Clostridium, like freely available microorganism in the soil, that degrade the dead plant material and convert into amino acid. Amino acid convert into ammonia, that we consider as the ammonification. Okay, now the ammonia is produced. Ammonia is produced. Now this ammonia and you know, the same bacteria by not only same bacteria, rhizobium like nitrogen fixing bacteria, again nitrogen gas මෙයාලත් උදව් වෙනවා. Nitrogen gas in the atmospheric nitrogen gas, N2 gas, directly convert into NH4 and NH3, ammonium and ammonia, NH4 and NH3 by this bacteria and this bacteria plus rhizobium plus this freely available microorganism. Then you can see finally it is produced lot of ammonia and ammonium mixture. Now understand, now this ammonium and ammonia mixture, NH3 and NH4 produced in this process convert into NO2 minus, that we called as nitrite. These words are very important. Nitrite. nitrite. That's doing by nitrosomonas. Remember this bacteria very well. Nitrosomonas. Nitrosomonas is the bacteria. Nitrosomonas. Nitrosomonas. By these nitrosomonas bacteria, what they're doing, there's a special bacteria, ammonium and ammonia convert into nitrite. Okay. Now, this nitrite, NO2 produced by this nitrosomonas convert into NO3 minus, that is the nitrate, by nitrobacter. Okay, they are the microorganism involved for the nitrification. You can see different sources, the nitrogen coming from different sources, from atmosphere, from plant and animal dead parts, finally convert into nitrate ions. That is the process of nitrification. Understood? Quickly. That is the process of nitrification. Understood? That's how. That's not only nitrification. There is additional industrial fixation and atmospheric fixation need to add. But that's a full microorganism involvement for the nitrogen cycle. Are you good? Understood? Quickly, we have bulk part to discuss today. Still, we are in agriculture. Any doubt? Any process doubt? Amar, understood? Deneth? Okay, quickly note down this in the tute. Quickly note down. In the tute, I have given space. Space के लिए आगा ना टक्कर. Above part, okay, nitrification. Nitrification, above part, page number 80. Nitrification. This is the nitrification microorganism involvement. Complete. Quickly write down. Okay, I will zoom out this. Oh, what? This is the nitrification. Okay, you know the nitrification is one step. This every process happening in the nitrification. There is another process, denitrification. That's everything. This every process doing in aerobic condition. Remember, this every microorganism act. This Acetobacter, Anabaena, Clostridium, Rhizobium, Nitrosomonas, Nitrobacter. Every microorganism act on aerobic condition. Aerobic condition, only aerobic condition. Aerobic condition. If it is anaerobic, the story is different. If it is anaerobic condition, it is NO3 nitrate ions convert into again nitrogen gas in anaerobic condition. Anaerobic condition. Then write down example. Pseudomonas, Paracoccus, like microorganism species that doing opposite way, that is anaerobic condition, that is the denitrification. That's the denitrification. Remember, this above process is nitrification. I have given both. Then the lower three rules, the bottom three rules, you can write down. The denitrification, this is the red color one is the denitrification. This is nitrification. This is denitrification. Yeah, the Clostridium also, the Clostridium not common one, the Clostridium also, if it is the Clostridium also working in anaerobic and aerobic both condition. In anaerobic condition, the Clostridium also involved for the denitrification process. It's not common. Remember these two examples. Not always. Okay, 9:20. Do you need break? Or can we continue? I have more about the agriculture complete. How we use microorganisms in food industry. Now we're going to discuss about food industry. Next, yeah, I'll give you break. Then I will explain how you use microorganism in beverage productions, like beer production, wine production, like beverage productions. Finally, in dairy productions, and dairy production and compost and biodiesel productions and bioremediation. Okay, and how we use microorganism in wastewater treatment. Okay, then take a break. And how many minutes do you know? 9:45 we start again. 9:45 I start on time, join on time. 9:45. Okay, we discussed about how use microorganisms, how important in agriculture. Completed, no? Actually, go to the second one. How microorganisms use in food productions, food and beverage productions. Okay, go to page number 19. Okay, food productions. In food production, first of all, we're going to discuss about bread production. I already explained. I simply explained the bread production. What are the main raw materials? What is the main raw material in bread production? What are the main raw material? It is wheat flour. Wheat flour. First of all, in bread production, we need to prepare a mixture of bread production, adding water. The mixture of wheat flour, adding water, and wheat flour plus water. And additionally, we need to add some sugar amount. Sugar amount as a substrate of the microorganism. I explained earlier in the question discussions. Therefore, I will not take more time for the bread productions. In the sugar applied for the substrate for microorganism, and we need to add Saccharomyces cerevisiae or yeast microorganism. Saccharomyces cerevisiae. We need to add some yeast amount. That means yeast is a fungi, and the fungi amount introduced this mixture. Then what happened? This Saccharomyces cerevisiae, I told, release special enzyme. What is this enzyme? The enzyme is invertase. Invertase. Then we added sucrose. What happened? Sucrose convert into glucose. Glucose plus fructose. Building blocks. By invertase releasing invertase enzyme. Remember, invertase enzyme. Invertase enzyme, I already explained. Invertase enzyme. Invertase enzyme. Invertase enzyme. Invertase enzyme. Now this glucose, now the glucose is a good surface for this Saccharomyces cerevisiae. Then start the fermentation process. This produce glucose convert into by the fermentation process. That means we need to give some favorable condition for microorganism. We need to cover, and for fermentation, you know, the Saccharomyces cerevisiae more preferable. It is faculty to anaerobic, more preferable to live in anaerobic conditions. Anaerobic conditions. Therefore, we need to give favorable condition for this Saccharomyces cerevisiae. The favorable condition is, if it is anaerobic condition, it's better. Okay, it's better. Then by this Saccharomyces cerevisiae, I am writing easily yeast. By this yeast, what they producing? C2H5OH. C2H5OH. That means ethanol and carbon dioxide. C2H5OH and carbon dioxide. This is ethanol, and carbon dioxide gas. Okay, after this incubation or fermentation period, you can make a PT mixture hadala. After that, we need to give some fermentation incubation period. In this incubation period, what happened? In this incubation period, this ethanol and carbon dioxide is producing. Ethanol and carbon dioxide, the sucrose amount, we added sugar, convert into ethanol and carbon dioxide. What happened? The volume of the mixture. The volume of the mixture is increasing because this carbon dioxide, all carbon dioxide cannot remove environment. They cannot release to the environment. Therefore, some carbon dioxide particles remain inside the mixture of starch mixture. Therefore, the volume of the starch mixture is increasing. The starch mean the wheat flour mixture is increasing. That's the process of incubation. After incubation period, we need to convert the mixture into small particles, and we need to fill into molds and dry in the oven. Okay, in the drying process, what happened? We increase the temperature. While increasing the temperature, what happened? This ethanol is evaporated and carbon dioxide is expansion. The carbon dioxide is expand. That's air. It is when temperature increase, it is expand. The air particles expanding. Due to this expanding, the inside the structure of the bread, what happened? It's the texture is coming. The smooth texture is of the bread is coming due to this carbon dioxide expansion. Okay, that's the process of bread production. That's, I think, third or fourth time I explained the same thing. Yeah, what's the question? Will the dough still ferment even if we don't add sugar? No, don't add sugar. The starch also contain very small amount of sugar. Sugar දැම්මේ නැතත් it is small amount of fermentation doing, because the starch also contain very less amount of sucrose, sugar. Then it is the yeast converting this utilizing this glucose, and it's converting to ethanol and carbon dioxide. But if we add this additional sugar, we can speed up this process. Amma, we understood? The sugar is not we adding to otherwise breads will be sweet like buns. Okay, we very small amount we need to add for only for microorganism activity. Only for microorganism stimulate the microorganism activity. Very small amount we add. The starch also contains some amount of sugar that also you can utilize by the microorganism, but additionally if we add small amount, that is an additional advantage. We can speed up this process. Speed up this process. Okay. Okay, that's the bread production. Okay, this amino acid production process actually you can read amino acid, different amino acid I have given here. For different amino acid, different microorganism we can use. Lysine and methionine, monosodium glutamate, like amino acid, different microorganism we can use. Okay, better to remember, make a short note of this each amino acid and that producing microorganism. That's only you need to remember. It is possible to come a MCQ question. Okay, now I will directly go to the use microorganism in alcohol beverage production, because I 10:30 I have another work. I need to complete at least beverage productions before 10:30. Okay, I will explain beverage production. You can make a short note in amino acid production. Okay? Beverage production. Okay, in beverage production, first of all, we need to understand the 2D production. 2D production is same as this ethanol bread production, the same process. For 2D production, you can practice this in your home as well. For 2D production, we need to first of all we need to get the mixture of sucrose. For sucrose, we can normally be getting the sucrose solution coming from kitul coconut like plants. That mixture we called as that's a sucrose solution. Okay, sucrose solution, it rich in sucrose. It's a make it කියන්නේ කොහොමද? I have given the tune, no? Cutting inflorescence of plants such as coconut and palm trees, commonly mira කියලා තමයි කියන්නේ. Mira. This is a sucrose solution. This is a sucrose solution. Cutting the inflorescence of the palm tree like coconut, kitul, then it is the secrete juice parts we can collect. After that, we can add, this is very simple process, we can add Saccharomyces cerevisiae. Saccharomyces cerevisiae yeast. Okay, yeast. Then what happened? This Saccharomyces cerevisiae, sucrose, as I explained, sucrose is contained here. Sucrose convert into glucose by invertase enzyme. Glucose and fructose. This glucose again convert into by Saccharomyces cerevisiae yeast. This glucose convert into C2H5OH. C2H5OH is ethanol plus carbon dioxide. This carbon dioxide releasing, then you can see the ethanol is producing. Do you know what is the ethanol percentage of 2D? Ethanol percentage of 2D? 2D කියන්නේ මේ රා කියලා සිංහ. රා කියලා කියන්නේ What is the ethanol percentage? What is the ethanol percentages? What do you think? Normally, it is very low ethanol percentage because in this Mira sucrose solution, the inflorescence solution, it is sucrose concentration is very low. Therefore, it contains lot of water. Therefore, the producing ethanol amount only 5 to 6%. Remember, 5 to 6%. Normally, it's average 4 to 6% actually. 4 and 6, at least like 5%. Averagely, it is like 5%. Very low amount of ethanol. The other 95% is water. The water is the main ingredient in this inflorescence solution. Okay, now that is the 2D. Very simple. Okay, now actually this process very simply you can do in your home. Very simply you can do in your home. Okay, you can take a glass of water. Glass of water. Hari කරලා බලන්න. Glass of water and add 2 to 3 sugar spoons. Sucrose, sugar. Then mix well and keep in environment 2 to 3 days. 2 to 3 days. Make a nikantian. No, don't need yeast. If you want, you can buy a small amount of yeast and you can add. If not, if you don't have yeast, you can keep in the environment. Okay, if you keep in environment, what happens? The environment available microorganism, this is a sucrose solution is a good surface, good substrate for this microorganism. Therefore, environment available microorganism, they come and grow on this surface, on this mixture. Therefore, what happened? Start to fermentate. Then ethanol and carbon dioxide is produced. Ethanol and carbon dioxide is producing. Carbon dioxide with bubbling make it bubula dana wage pena. That's mean carbon dioxide releasing. You can practice this. Okay, you can do this by the coconut water. Coconut inside water you can use for this process, because it also a mixture of sucrose. Okay, you can take coconut water and put into a glass and keep open 2 to 3 days. 2 to 3 days. It is actually after 4 days you can see its color is white color. Now for coconut water, no more color, no specific color. When it is 3 days, after 3 days, it is the color is converted to white color, and it's the smell is totally different. You can try. Okay, you can try, and next week tell me what's your observation. You can get sucrose solution, that means sugar solution, or coconut water or king coconut water also okay. If it is coconut water, I think better, because it has more sucrose level, more than others. Coconut water best. If you want, you can get coconut water and additionally add sugar, no problem. Take you can practice different way. Actually, you can do experiments. For first one, you can get you can practice. For this one, you add only water and add sugar. Only 3 spoon of sugar. 3 spoon of sugar. 3 spoon of sugar. For next one, next class, you add only coconut water. Coconut water. For next one, you can add coconut water and sugar both. coconut water and sugar. So additionally, if you can find yeast, you can add another experiment. Take coconut water and sugar and yeast added one and without adding. You can practice actually. You can ask from hardwares the yeast කියලා ඉල්ලන්න as yeast they know. It is very no more expensive, very cheap. The yeast is very cheap. The small amount is required because when we adding, this is a fungi type, it's not a chemical, it's a microorganism, it is a fungi. Therefore, when they have favorable condition, what they doing, they growing and reproductive rapidly. Therefore, we don't need to add number of spoons. 1 millimeter, 1 gram is enough. Oh, very small milligram amount is enough. We need to only introduce, then they doing their job, they reproducing and they increase their population. Okay, can you practice this? Okay, that's 2D production. Then the wine production and beer production is, let's do next day. Now I have 10:20. I have a special work at 10:30. Okay, let's do in tomorrow. Tomorrow you have practical. Tomorrow can we get 2 hours? From next week you have practicals. Tomorrow you don't have practical. Can we discuss this lesson 2 hours tomorrow morning? We start your practicals from next Wednesday. No, tomorrow, no, evening. Tomorrow evening, our 26th class. From next week, every Monday, you have practical sessions. Morning, 8 to 12. But tomorrow don't have practicals. It is not finalized yet. From next week, we start your practicals. The practicals mean the first practical part actually easy. The first practical part, the measuring instruments. The mid part is important. Heat lesson practicals, the force lesson practicals, they are important. They are commonly asked in the exam. And mechanical properties practicals. Okay, I will give you full practical guide and discuss all practicals. Okay, can we get tomorrow 2 hours for this remain part? If anyone can't send me, then 9. Okay. Then we start 9 to 12. Try to do maximum in cell structure lesson. I try to cover maximum in cell structure lesson. If all productions we can discuss, and if I can cell plant and animal tissues, stem tissues and leaf tissues lesson, if we can complete, we can discuss. Then the cell structure lesson 75% is complete. Only animals and birds part is remain, and tissue culture. Okay, then it's complete. Okay, you can practice this one. Okay, you only need to sucrose solution. And why it is keep when it is without adding yeast, when we keep the solution in environment, in open environment, how it is converted into ethanol. How it is converted into ethanol? Yeah, I told earlier. The environment contains a lot of fungi. Therefore, this fungi act on the surface because it is a good substrate. Therefore, they can grow on the solution, then convert into ethanol. But it is not a safe method. It is not the safe method. But the reason is we don't know which type microorganisms go in here. Therefore, it is not suitable for use. Drink, because the same structure, the ethanol is okay. Ethanol is okay, but the methanol, if it is produced methanol, you already know methanol where we using. Methanol used in biodiesel production. The ethanol and methanol, their taste, their color, all are same. The ethanol is C2H5OH. The ethanol is C2H5OH. C2H5OH. The methanol is CH3OH. CH3OH. But if they are chemically different, their structure, their color, taste is same. Therefore, it is most possible to produce methanol. The methanol, is it favorable for us? CH3OH, methanol. Methanol is highly toxic compound. එතකොට environment available microorganism, it is dangerous, but you can practice, but don't try to taste. Okay, it is possible to produce methanol. The methanol is highly toxic. Okay, that's all about 2D production. You can practice in your home, and next week I ask observations. So next week tell me observations. Okay, tomorrow at what time we start? 9. Okay, 9 a.m. to 12 p.m. We try to cover maximum in cell structure lesson. From next week Wednesday we start your practical components. Understood? Others, are you okay tomorrow? Okay, tomorrow we'll do. Amar, you only have class, I know. You can watch recording evening. I will upload the recording. Watch and complete the cell structure, because the cell structure, if we keep the cell structure, it is a headache because it is a bulk lesson. If we complete, then we have only the electronics and environmental chemistry. Then we can complete within this month, we can complete all. Then only one lesson within this week, this month, we need to try to complete environmental chemistry up to. Then only electronics is no theory lesson. Then we can complete. We have another 2 months. Therefore, do your maximum within this month and next month. Okay, don't keep anything in our in my side. I am doing maximum within this month and next month. Try to do your side, your maximum to try to complete everything within before final month. Then it will be very helpful for reading for your exam. Okay. Okay, now 26. I have to stop now. Tomorrow 9. Okay, 9. Okay, good night.